Thank you to the organizers for inviting me. I am going to talk about the effects of pre- and postnatal exposures on neurodevelopment. And I'm going to start by introducing you to what MEG is. So magnetoencephalography is very similar to electroencephalography, which people are generally more familiar with. But what we are measuring with both EEG and MEG is the excitatory postsynaptic potentials that happen come from the apical dendrite of pyramidal cells. They are nicely aligned, which allows us to measure the magnetic field that comes from those currents. And we can measure those magnetic fields outside of the head. This is the instrument that we use. And inside this plastic helmet, we have a bunch of sensors that are located around the head, and that allows us to measure the magnetic field pattern. And from getting that magnetic field pattern, then we can figure out where those currents are coming from in the brain. So this is just a depiction of that. And we also, similar to EEG, have high temporal resolution, so we can look at the location as well as the timing of that activity. So just as a brief overview of the difference between fMRI and PET, for example, the other non-invasive neuroimaging techniques, EEG and MEG, have higher temporal resolution, so we can collect data at about 1,000 hertz or every millisecond. Other invasive measurements also have this type of high temporal resolution, but EEG and MEG are the ones that can provide it non-invasively. So this is just an example of why that timing is important and useful. So this is actually the onset of a seizure. So you can see the spike here, and then it propagates. So each of these lines is a different EEG channel. But you can see that the activity within a half a second starts propagating to other parts of the brain, and then within another half a second, it is propagating to the full brain. So the reason that we use MEG and not just EEG is because MEG actually provides better spatial resolution. So one of the clinical uses of MEG is to map epileptic activity. So that's what this is denoting, is epileptic activity that was measured during MEG, and it maps on nicely to what was identified as the ictal zone using intracranial or invasive EEG measures. If you are interested in the clinical use of MEG, there is a nice topic special issue on this in the Frontiers. But what I'd like to move on to is the research that I've been focusing on for quite some time now. So one of the questions that I ask in my research is looking at the early identification of neurodevelopmental disorders. One of my studies was looking specifically at autism spectrum disorders. So this was looking at the auditory evoked response. And this was in fairly young children. This is age in months. So this is a 6-month-old down to a 46-month-old. And then the kids with autism spectrum disorders are a different time frame, but these are just specific examples. What we're looking at here is the timing of these peaks. And what you can see in this finding is that for the different peaks, this is peak 1, 2, and 3, that in our typically developing children, then the latency decreases with age. This is quite well known, but what we found is that with the individuals with autism, their latencies, and this was a cross-sectional study, but the older kids with autism had delayed latencies relative to the typically developing controls. So again, going back to our motivation to try and look for early markers of atypical brain development, this doesn't really work out that great because there's overlap in this age range when you can already identify kids with autism behaviorally. But there are still differences in the developmental pattern. So another topic that we've been focusing on quite a bit actually is looking at the effects of prenatal alcohol exposure. So there's quite a bit of preclinical research happening here in the neurosciences department in rodent models looking at the effects of prenatal alcohol exposure. And we do think this is important clinically as well because the prevalence is high. So fetal alcohol spectrum disorders is the disorder if you have sufficient cognitive or behavioral effects due to prenatal alcohol exposure. Diagnosis is hard unless you have facial dysmorphia, but this does not account for most of the cases of individuals with FASD, and life outcomes are poor. So we looked in infants, trying to understand the neural dynamics. So similar to EEG, this is what MEG signals look like. And you can identify different periods of neural oscillations, and that's what we had done with this particular study. This was looking at periods of rest in infants while they were sitting there in the scanner awake. What we found was that the kids with prenatal alcohol exposure had increased spectral power in the different frequency bands. So delta, theta, alpha, beta, gamma are different frequencies. And we also found that there was a dose effect. So with moms who had consumed more alcohol, then there was stronger power. So this does seem to indicate that there's a chance for early identification of atypical brain development. And this study was done at 6 months of age. However, the challenge with looking at only kids with prenatal alcohol exposure is that we don't actually know if they have the cognitive or behavioral effects related to FASD. So the PAE kids, not all of them are expected to go on to have an FASD. So we've also been doing studies in older children. So this is now a study in 8 to 12-year-olds and looking at the same different frequency bands. You can see, and now the FASD group is broken up into 2 subgroups. So the AR&D, which is alcohol-related neurodevelopmental disorder, and then the gray is the kids with fetal alcohol syndrome or partial fetal alcohol syndrome. And it's quite different than what we saw at 6 months of age because mostly there were no differences by group, except for only in this one subgroup did we see a reduction, in contrast to the increase in power that we had seen in the prenatal alcohol exposed kids. So we know that things are changing, normalizing, which is good. But there are still some abnormal neural dynamics within the brain. We're also looking at functional connectivity using the same resting exam. And we can look at the functional connectivity in different brain regions. And using dynamic functional connectivity, you can essentially identify different states that the brain is in and see if there are differences in how the brain is evolving over time. And there are many different metrics that you can get from this, but one of the things that I want to focus on is this change between states. So that was significantly different, and the kids with FASD change states more frequently than the typically developing controls. So this is consistent with what we see behaviorally because these kids also have attention deficits similar to kids with ADHD. So you may expect that the brain is essentially switching between states more frequently because of that. So we do find it important to continue to also look for both structural and functional links. So alpha oscillations, going back to one of the physiological frequency bands, is associated with attention. We know that kids with FASD have attention problems. And we also know from preclinical literature that alpha oscillations are generated from thalamus. And so we looked at the cortical thalamic connectivity in these kids with FASD to see if the structural, the fractional anisotropy specifically measured with diffusion tensor imaging was different in the kids with FASD. And in this case, these different tracks cortical thalamic tracts showed reduced fractional anisotropy in the kids with FASD. And that is now across the whole group rather than just in the subgroup of kids with FAS. And we also found an association between the fractional anisotropy value and alpha power, such that if you have increased fractional anisotropy, then you have increased alpha power in the direction that you would expect. Although this association was stronger in the typically developing controls than it was in the kids with FASD. We do have a new center grant where we're collaborating with our preclinical collaborators to better evaluate what's going on in terms of changes in myelination, as well as this cortical thalamic connectivity. So there are just some other postnatal variables that we've been looking at. This was the developmental connectogenomics study. This is a multi-site study measuring MEG in 200 kids. These are just some of the demographic variables across the two sites, so New Mexico and Nebraska. But we were, again, looking at the association between alpha oscillations and attention in this study. And we did find that kids with better attention had higher alpha oscillations in the frontoparietal network, which is really the cognitive control network that we'd expect to be really linked to attention. And the last thing I want to talk about in this population, this is the same cohort, is we asked a trauma questionnaire. And so we were essentially just asking how many different types of traumas have you been exposed to as a child. We did not include abuse and neglect in this questionnaire, but we were focusing on other traumas such as neighborhood violence and natural disasters. So this is an important thing to look at because we know that exposure to adverse childhood experiences and so on and so forth lead to increased prevalence of mental illness in adulthood. So we did find structural differences. So this was, again, looking at fractional anisotropy. We identified different regions of interest based on the literature in other individuals who have experienced trauma. There were quite a few behavioral differences. So anxiety, anger, depression, dissociation were all quite different between the low trauma and the high trauma group. If we did a median split on the number of traumas they were exposed to. And from this original region of interest analysis, we found that there were 5 regions that were significantly different. And in all cases, the fractional anisotropy values were lower in the individuals with high trauma exposure. These same kids also went through a go, no-go task. So this is similar to what Kent Keel was talking about. In this case, we were looking again at the MEG response, and we saw both changes in amplitude, which was reduced amplitude, as well as changes in latency. So this is how quickly does the brain respond after seeing this stimulus. And in this case, the high trauma group actually had faster latencies. So this may be related to hypervigilance, for example.
Just letting you know we have 2 minutes.
Okay. So my conclusions, so be on time. We are continuing to push for more complete understanding of the effects of both prenatal and postnatal exposures to improve outcomes for children. We are examining the neurodynamics. We'd like to examine neurodynamics in more realistic scenarios to get a better understanding of brain function in everyday life. And then our long-term goal is to continue to develop improved methods for early identification and intervention for neurodevelopmental, neurological, and psychiatric disorders. And we have funding from both NIH and NSF for this work, and I'd like to thank my collaborators who have worked on these projects.
Thank you very much.
Questions.
Okay, do we have questions? Do we have any? I have a question actually. In terms of the results that you showed on attention in children and looking at the power analysis, do you envision that could be used for drug development or for biofeedback for children for therapy? Yeah.
Yeah, I think I haven't spent so much time looking at drug development, although there are some studies showing that certain drugs have influences, obviously, on different neural markers, including neural oscillations. I think biofeedback is something that people have continued to work on. It has not really taken off yet, but and then we have a music study currently to look and see if you can use things like music training to improve brain function and some other types of neurostimulation, for example.
Thanks. Yes, I have a question also. Thank you very much for a lovely talk, Julia. This is Chris
Thank you.
Leonard. I had a question about the kids that you had looked at alpha wave activity in which they may have been exposed to fetal alcohol or alcohol during their fetal stage, and you mentioned that there were similarities with kids, older kids that have ADHD, and I just was hoping you could clarify that a little bit. In other words, do you see the same changes in those ADHD kids, or are there differences between them? Thanks.
Yeah, so we haven't looked at an ADHD cohort specifically. We know that kids with FASD have attention problems. So there's definitely overlap with the ADHD diagnosis, but we also know that they respond differently to stimulant medication. So oftentimes stimulant medication is not effective in kids with FASD. So we do think that the underlying mechanisms are different between sort of a more pure population of kids with ADHD versus those who have attention problems because of prenatal alcohol exposure.